All right. Not that I need you to know all my problems, but this has a couple of weird things in it, and I can't figure out exactly where to put them. So I'm just going to do my best. That's all I can ask. Okay. This is gene regulation. There's epigenetics in it, but it is by far not the biggest piece. Okay, we are 3 units left. This one and 2 more. So, and the 2 more are super familiar. Well, I say that. Super familiar and super easy. So that's nice. Natural selection and ecology. So this one is probably our last one that is has stuff in it that this is also our unit 2. So we are way back to the beginning of when we started. And so there's going to be some stuff in here that you remember way back there, but maybe not well. So this one actually might be one of the more helpful reviews because it's stuff that we can pull back out from basically September. But this is gene regulation. Like I said, we did this back in Unit 2. This was the second thing I do. I feel like it's one of the most important units of biology because it's how DNA works. And it's how DNA makes proteins. And it's how all of that is regulated and how everything that we've been talking about, genetics and all that stuff, how it works. And so, but the problem is that this unit randomly, our unit didn't, but this unit randomly also includes DNA replication out of the clear blue sky. And so it's kind of like, where am I supposed to put that in gene regulation? So I'm just going to stick it over here and we're going to do it first. We're just going to get it over with. Okay, so it's going to go on the left here, and I'm not going to go over every detail. Once again, this is a review, not a reteach. And so if it's something that you're like, well, I don't really remember exactly that, then you can go back and review maybe a little more in depth. In DNA replication, they also revisit the structure of DNA. So make sure that you know all the ins and outs of DNA. We did that, I think, when we first started on the unit 1 or something, when we started reviewing. So that would have been like last Monday. But in here, it did make note that you are supposed to know purines versus pyrimidines. So, and that was something that I said, well, it's not here at the structure. Well, here it shows up in Unit 6. So great. So maybe if you have with you all of your sketch notes, you might want to page back to Unit 1, which would have been like last Monday. So this is Unit 6. So 5 pages ago. Pretty sure it was Unit 1. Let me see. They have been messing with the standards so much that they've taken out stuff and put stuff in. So sometimes they catch me unawares. And I'm like, oh, never mind. That is still in here, just way back here. I think it is Unit 1. We should have talked about biomolecules. We should have talked about DNA, nucleic acids. We should have had A, T, C, and G in there. Just know that your G and your A are purines, if you want to mark that down somehow. Purines are the double ring. So like when I've been drawing, like ATP, I drew that little thing look like a little, I don't know. Like they're not really rings, so I don't like making them completely circular. They're kind of funky looking like that. Like that, whenever I drew that and then it was attached to your nucleotide there, I did 2 rings. If I left one off, then that would have been a single ring. So purines, which is spelled like that, by the way. I'm going to erase all this. Don't write this on your notes for today. Purines are A and G, and pyrimidines are the rest. Okay, so there's 3. Purines. God. This is purines as a double. And then pyrimidines would just be a single ring. And that's just talking about their structure, like how they're actually formed, like in a chemistry way. And pyrimidines are single ring, and they are thiamine, cytosine, uracil. Okay. And one double is paired with one single whenever you're doing bases. Okay. So I think somewhere we wrote down the bases. Just make sure you know which one's which. I guess I'm going to say you probably won't even have to know, but just in case, I have some stupid way to remember it. Like, I don't know, purines, gap,
pure gap, and
pyrimidines.
You say cut with that.
Cut the pyrimidines. I just put it together like cut pyramids, and I don't know. I just have like a piece in my brain somehow, and it's no great way to remember it. If you find some better way to remember it than pure gap, I don't know. It's such a yuppie thing. And then pyrimidines, I think that's like cut, like little pyramids are cut. I don't know. It's just ridiculous. But anyway, I just have a little memory trick that I use, and that's how I remember. And then purines are double and pyrimidines are single. I think of a pyramid as being like more narrow than anything else. Okay, so yeah, that's a dumb way to remember it. But anyway, okay, cool. So that was supposed to be in there. The rest of it is the structure that is included here in Unit 6 is stuff we've gone over before, like prokaryotes have a single and single ring structure, and eukaryotes are paired in their linear and all that stuff, all the stuff we've been through. But let's talk about this. Okay, so I'm just going to draw just a little abbreviated DNA that's open to be replicated. Okay, whoa, hold that ugly real fast. Okay, so this is our little DNA, and I'm not going to draw out every step, and I'm not going to tell you what everything does unless you have one particular one you just want me to remind you of. If you have somewhere you can find what they do and stuff, but this to me, it's just like, let's get the major parts. So we're going to go that this template strand right here is 5'3". So I got to follow it down here and figure out which end is the 3 end. And this one is 3 to 5. Yep, that's right. Okay. And so first of all, we have to remember that DNA is replicated from a 5' to 3' direction. That is the way the polymerase works. It can lay stuff down 5 to 3, and DNA has to be anti-parallel. Okay, so with those two pieces of information in your head, you should be able to identify the leading and lagging strand. Okay, the leading strand, the one that goes fast, and the lagging one that goes slow. Okay, so if I have to lay it down 5 to 3, and it has to be anti-parallel, so if there's a 3 here, it has to be a 5 there, then this one is my leading strand. Okay, and this one is my lagging. Now, there's about 3 different ways that I've seen them put a diagram on the test and wanted you to identify which one was replicating correctly, okay, which image is correct. They have done it just by putting numbers on, like this, like 3', 5', like that, and you're supposed to say which one it is. So if your template strand is 3 to 5, because, oh, by the way, this is a replication fork, so we're moving in this direction, if that's confusing to you. Okay, because it's open here and it's not open here yet. So if your template strand is 3 to 5 here, then that's your leading, because you're going to lay down bases, or the DNA polymerase is going to lay down nucleotides in a 5 to 3 direction. It'll just be able to go do. I have also seen them put arrows, like the one is being laid down like this, and then they'll put arrows. Well, actually, if you wanted to do it correctly, you'd start here. You'd say that, then you'd say that, and then you'd say that, and do it in little Okazaki fragments. Okay, so I've seen them do, and this needs to be one continuous. I didn't quite make it long enough. I've seen them not have the numbers on, but instead just have these little arrows like that. And they'll give you 4 different, they gave you 4 different pictures in that one, like your multiple choice. The question was, which of these demonstrates DNA replication correctly? And then they gave you 4 different options, and they had like, this one being like discontinuous, and or both of them going this way, the arrows going this way and stuff like that. Only one of them did they have one arrow, and there was no numbers, so you just had to look at arrows. Only one of them did they have one continuous arrow going towards the replication fork, and the other side had these little discontinuous arrows going away. So some of them had both discontinuous, some of them had, both continuous, some of them had both arrows pointing towards the replication fork. So I've seen that. I've seen it with the numbers, and they want you to identify which is which. A couple of different ways, but it shows up, not every test, but pretty frequently, for you to be able to identify how DNA replication, like which way it goes. Okay. All right. So questions on that, and that's basically, if you'll remember, the carbon numbering on the little sugar. So that's what that means. That little 3' is like, that's the 3rd carbon on the little house, the little pentose. And then the 5th one is the 5th carbon. So, but honestly, we've drawn that several times, and I haven't ever really seen them focus on that particular portion of it. It's more the big picture. Okay, questions on that? All right, so what else do you have to know about replication? Well, you need to know that it is semi-conservative. So here's a little list of what you need to know about it. So it's semi-conservative, which means, in case, I'm not going to write a definition, but in case you forgot, that means that in the end, what you get is part template, part new, right? So you have 2 DNA strands that are part template and part new. So the two new strands being built don't go together. That would be like fully conservative, because you'd have like the new ones together and the old template would be completely conserved. Not that, okay? But it's not like you toss the template away, completely, and you have new DNA. Is that you have one template strand and one new strand that are bound together, and that makes your DNA. Okay, so that's semi-conservative. You need to know the enzymes. So this is your little list of need to knows. You need to know the enzymes. Okay, so we have helicase. I'll list them for you. But this is one I'm not really going to go into detail with them unless you just need me to. See what you can remember. I better call it DNA polymerase. Picky about that. And they should be. There's an RNA polymerase too. So there's helicase, topoisomerase, DNA polymerase with an E at the end. Oops. And primase and ligase. And those are your enzymes that you're supposed to at least be familiar with and kind of know what they do. Know it for sure, know that they are in DNA replication and not in transcription, which is coming up next. Okay. All right. And that's it for DNA replication, unless you have a specific question, which I'm happy to answer if you do. Yes.
I'm just having a quick question. There's rRNA, tRNA.
Yes, and we'll get into those right here.
Okay.
Yeah, because we're about to do transcription translation, which encompasses all the RNAs, all 4 of them. So, okay. Coming soon. Anything about, that's why it's kind of funny that DNA replication is in here, because it's like, it doesn't have anything to do with like the big picture, which is transcription translation is kind of the big part. All right, any questions about that? Okay, cool. All right. Excuse me. So the rest of our paper will be dealing with transcription, translation, and the regulation thereof. Okay. So I'm going to try to put it, this is where I kind of can't figure out exactly. I'm going to try to put transcription like kind of largely here, and then out to the side over here, I'll put what regulates transcription. Okay. And then I'm going to try to put like translation down here, and then kind of what regulates, or what's in between them, even like what's in between regulation and what. So it's going to be kind of like regulation over here and the process in the middle. And I'm going to try to feed them together. This is where I was like, I can't ever quite get it right. And I've drawn this enough times that I should be able to. Okay, so first of all, this is our central dogma, or you'll see it called a few things. So I'll give it a couple names here. So central dogma of biology basically means this is the main central point of biology. You can call it protein synthesis. Okay. And it has 2 parts. It has transcription and it has translation. Don't get them confused. The names. Okay. If you can't remember the name of it, don't use the name of it, just describe it. Like mRNA being made from DNA. Like if you can't, in the moment you're like, transcription, translation, oh my gosh, don't say the wrong one. Okay. And if you just absolutely cannot remember, just say when DNA is producing an mRNA strand off of its coat, something like that. And it takes longer to say it, but it's better than saying the wrong thing and missing the point. Okay, because we're not usually after the vocab. But if you can, transcription comes first. Sometimes people say, well, they're in alphabetical order. Transcription, translation. The C comes before the L. If that helps, I think transcribing is like writing. So you're writing the DNA code onto mRNA. It's like you're actually writing it, and then translating it, you're turning mRNA into something completely different, a protein. And so that's kind of how I remember it. But sure, if alphabetical order helps you, whatever. Just I have seen it too many times in free response questions where a student wrote the wrong one, and you can't award a point for the incorrect vocab term. Okay, so first up, our step 1 is transcription. And this is DNA to RNA. So DNA to RNA. We have a difference between prokaryotes and eukaryotes here, in that prokaryotes don't have a nucleus, but in eukaryotes, this occurs in the nucleus. Okay, we're going to have a couple of different differences between, which is another point of depth in this little sketch notes. It's kind of hard, is how do you differentiate prokaryotes and eukaryotes? I'll try to do it concisely so that it's not a huge mess. But so in eukaryotes, this is going to happen in the nucleus. It's because DNA cannot leave the nucleus. So anything that happens with DNA must occur in the nucleus in eukaryotes. So that is tested all the time. Okay, all the time. It's like location. Where do you expect this to happen? Where do you think like so this happens with something called RNA polymerase as the enzyme. So where would RNA polymerase be found? You know, stuff like that can pop up. You have to, well, RNA polymerase is going to be found in the nucleus because RNA is being made in the nucleus because that's where DNA is. So, I mean, it's like you kind of have to put those things together. So basically what we're going to see here is we're going to see your base pairing rules, right? But the RNA doesn't actually bind to the DNA. It's just kind of making the little mRNA. So, and by the way, this is mRNA. So I'm going to just draw a little picture just to have a picture for fun. So if you want to draw a picture, you can too. But basically, instead of a fork, we have a bubble. Now you can also have a bubble over here, by the way, because they can be replicating the other way. So don't think just because you see a bubble, it's got to be automatically transcription. You need to look for stuff. And what you're going to look for is that an mRNA is being produced somehow. So I'm going to draw it coming out like this. Okay, so here's your mRNA, right? And so how you're doing this is your base. So basically, if you have a T, if you have a T here, of course, then that's going to be an A on your RNA. If you have an A here, that's going to be a U. If you have a G here, that's going to be a C. You can just make some little code for yourself. And then maybe all of this has occurred over here and you've already.
There's no T's in.
No T's in RNA, which is funny. You said that I just put one in there and was erasing it. I'm like, no T's. What are you doing this, little head? Okay, so basically you're just making a little mRNA on the DNA code. Now, why are you doing this? Because you need to make a protein, right? So you need to actually have a protein. You have been signaled from the outside world or from wherever that a protein is needed. That could be an enzyme. It could be a channel. It could be a whatever. You know, proteins do everything. And so a signal has come in and it has arrived in the nucleus. Think of our cell signaling process where it's like, oh, here's the reception, here's the transduction, and here's the response. Here's the response, okay? We're making mRNA. And so now that we're making mRNA, this is going to head to the ribosome here in a second, but we got to do some regulation of this process first. This is going to head to the ribosome and be translated into a protein, which is what the signal required, right? The signal was like, hey, we need some more gastric enzymes up in here or whatever, okay? So that's kind of what's going on here. Let me just check on what depth we have here. I have my whole list of crap. It's ugly. Let's see. Okay, vocab. Three of these is called what? What is three of these nucleotides? Codon. There we go. So codon. So this is a codon. I don't know where you have space, but that's a vocab word if you need to like mark your vocab words here. So this little box, so AUG would be a codon, and you use a codon table or a codon wheel to that. They'll give you a table, not a wheel. So I try to get away from using the wheels because we don't see those on our test. So a codon table, they will give you that. They don't give you an anti-codon table, so make sure you just use codons. What other vocab did they have for you? I don't think there's anything else as far as that goes, which is funny to me because they don't talk about DNA polymerase. I mean, they don't talk about RNA polymerase too much. Oh, they do. Okay, so this is going to be, they just don't highlight it. This is going, okay, so stuff you need to know. This is where I'm struggling. Like, where do I put this? This is done by RNA polymerase. So I'll just draw it kind of like encompassing it. And I'll color it in a little bit. Like this whole thing is a protein, a big fat enzyme. Okay, all around it. And it is RNA polymerase. So where in DNA replication, you have a whole smattering of enzymes that have to do the work. In RNA, when you're making a strand of RNA polymerase just does it all. Opens it, keeps it from, breaking, all those things at once. Okay. I hate this part. I don't even want to tell you that. I don't even want to say this. This is your template strand right here, right? It has a stupid list of words that they could use instead of template. And I don't know that they will. And I don't know why. They give you 3 other words that this thing could be called. And it's just kind of weird. They call it the non-coding strand. You're like, what? Why? Minus strand or antisense. Okay, I can tell you why, though it still kind of ticks me off. I would just call it the template strand if I were you, because that's easiest. So basically, if you think about it, what is being produced here, this mRNA, what's on the template is the opposite of it, right? So like if you have an A here, you have a T on your DNA. If you have a U being produced, then you have an A. So it's like the opposite. So that's why they call it the non-coding, because if you want to find the mRNA code, you're actually going to look up here. Like this is going to be the one that has the code that's identical to the mRNA, right? Up on the top. So this one is the non-coding. Literally hate that term. I think that is the dumbest biology term maybe that we have. It's like, now I'm totally confused. Thanks a lot. Minus strand, IDK. Antisense, because this one's the sense strand, I guess. Anyway, I am often irritated that they still have those words in your standards. Every time we get a new standard update, I'm like looking there going, are they gone? Because please, just call it the template strand and let's be done with it. Okay, cool. Any polymerase has to move in which direction? Which direction is it going to build the mRNA? This is a 5 to 3, this mRNA, yes. So it's built in a 5 to 3, just like the DNA is. Polymerase, that is a polymerase, like a limitation on the polymerase. So be it DNA polymerase or RNA polymerase, it's the same rule, okay? So it's reading a 3 to 5 and building a strand 5 to 3. Okay, cool. That's it for transcription, except for regulation of it. Everybody cool with that? Any questions there? Coming back to your memory, what I really just need you to do above everything else is to like put it all together. Like how I was saying, the signals coming in, you know, all of it. Like we need to pull all the biology together because that's going to get you the most points on the test, honestly, is that you can absolutely you can pass the test and even, you know, get a high 3-4 just knowing the details. But once you pull in the units together with the signaling and the evolution and all of the stuff together, that's when you kind of get your big bang points, like your 5s. So if you want to start kind of thinking, like, where is this coming from? Like, where are we in the nucleus? Like, okay, then think about the properties of the cell and the ribosomes. You know, I mean, it's like all of it just blends nicely. And I love that about biology. But it makes it like a lot of information that you kind of have to master all of it if you want to do super well on the test, which is challenging, but that's okay. Not many people hit that. About 8% and nationwide hit that 5, so that's fine. Okay, let's see. Let's finish out translation and then regulate it. Or do you want to regulate first? Let's regulate it. This is me. Okay, so this is going to be all of our regulation over here. There is a lot of regulation for transcription, and there's not very much regulation for translation. And there's a little bit in between, okay? So let's talk about regulating. Let's start here. Okay, so we're going to start with transcription regulation. So this is transcription stuff right here, step 1. So we're going to regulate it first. Basically, regulation of transcription involves turning it on. Or turning it off, I guess. But really, you're trying to, you know, most of the time, your cells aren't just going to be making mRNA willy-nilly, right? I mean, okay, cell differentiation, right? We've talked about this. We cannot have gastric enzymes being produced in our eye cells. Like, that's ridiculous. No one would think that was a good idea. Or anywhere else besides in the liver. That's the only place we want gastric enzymes produced, okay? So the template, the RNA template, the DNA template for gastric enzymes that's going to produce an mRNA is silent everywhere else. It is never turned on. So regulation of this process really involves, what do I need? What do I need in this cell? And when do I need it? Okay. This is not, once again, back to evolution. This is not an energy like cheap thing. This is expensive. It's expensive to make proteins. It costs energy to build this mRNA. It costs energy to make the protein. It's not something they just do, like have it turned on all the time. So you need things like promoters, right? You need things like methylation. You need things like histone acetylation. This is when your epigenetics comes in, as well as basically the whole like typical regulation part. So we start with just promoting it. Let's promote it. So you need to have a promoter, and your promoter is going to be upstream. Okay. When we say upstream, basically, you can see from my little picture here that we had to have a starting point in our DNA. Like there was a place, DNA is super, super long to the tune of like, number one is like, I don't know, what is it, 250, 246, that was pretty close, 246 million base pairs. That's long, okay? I can't even hardly fathom a million. Like if you were to say, how many is a million beans? I don't even know how big that is. But 246 million is ridiculous. And within that is thousands and thousands and thousands of genes, right? And so we just have to turn on the one gene that we need. We got to find that one. So they have a promoter, and so this is super long DNA, okay? And so somewhere along the line, there's a promoter that certain polymerase will recognize. And it's like, oh, it's my promoter. Here it is. So you can block the promoter, and it won't start transcription. Or you can let the promoter go, like leave it unblocked, and it will allow polymerase to bind and start transcription, okay? And so that's pretty much what a promoter does. It's upstream from the gene. And when I say upstream, that means this part like up here somewhere. So that I don't know where that would have met up, but like somewhere there's a place up here where the promoter, where the RNA polymerase, actually you probably have transcription factors that bind to it, okay? So we'll do transcription factors next. And these are proteins, because proteins do everything, might as well. And these work with promoter to start transcription. Okay, so basically what you've got here, I don't know how much space you have on your paper. What you have here is you have a sequence of DNA that is, sorry, my little video is off screen. You have a sequence of DNA that is a promoter region, Tata box, right? TATA, whatever. And it's a little bit different for each gene. So even though it's a combination of TATA, you might have a weird little repeats in different numbers of T's and A's and all that stuff. And it'd be like, okay, that's the promoter. And whenever the promoter is available, then the transcription factors, which are proteins, will bind in that area and basically recruit the polymerase. Okay, so they signal. And so then when all that happens, it goes. Okay, so with promoters and transcription factors, you're turning it on. Okay, you're turning on transcription.
RNA polymerase.
You're recruiting RNA polymerase so that transcription will turn on. So you're recruiting this thing in the background, which basically encircles the whole thing. You just don't want to color over the front. Okay, so that's your main one. That is the main way that we turn on genes. And like I said, and I'll say it again, we have all the genes in every cell in our body, right? Every single cell has all the DNA, all the genes. So we have to be very selective on which ones are turned on. Otherwise, you might just have eyes in weird places and hair, like your head hair might be growing out of your feet. I don't know. Because there's genes for that there, right? There's genes for that and the building blocks to grow hair, like head hair, out of your feet, like, or your liver. Hey, let's pick something even weirder, right? You could grow a whole ponytail out of your liver because the genes are there. But what we do is we don't turn them on. Okay, if we turn them on, then we grow the hair. Like it's chemistry. It's not like it's thinking, guys, this might be a bad idea. They don't think, right? So they actually just do whatever the chemistry tells them to do. Okay, so that means that there are a lot of questions on the AP biology test that will say something about genes, and they'll say like, which cells have the gene? All of them have the gene. It's not about which ones have the gene. It's about which ones have the gene activated, right? That's how you get the differences. And so that's a super important thing. Like it's like a basic thing that students will just glaze right over because it says which ones have the genes. And you're like, well, the liver cells have the genes for gastric enzymes. Well, so does every other cell in your body. It's just not turned on. Okay, so make sure that is solid in your brain, that we turn it on when we need it, but it's always there. Okay, that's why your cells are differentiated. That's how come your cells are different from each other. Yeah.
This might be too introspective, but if we have all of the DNA in each cell, what exactly allows us to identify each cell? Like what tells the cell?
Yeah, that is too introspective. Yeah, but there's some super cool videos that I can give you on human development that tells you in the very beginning, like when you were just a non-differentiated blob, like how your cells turn each other on and stuff like that. And it's really super cool, but it's really deep. It's beyond what the scope of our course. But yes, there are. So basically, it's signaling. It's all signaling. When one cell does this, because you have to, first of all, as a blob of cells, know which way is head and which way is butt. Like that's the first thing. And so it's kind of funny how that works. Lots of, we can get into it sometime because it's super interesting and fun, but probably not what we need right now. So, okay. But it's sometimes fun to think about how come your body knew that you were head up and butt down. Okay. So let me just review really quickly. So I am using my, standards today just to make sure I don't go over the top or I don't miss anything. And then this is what they say about stopping it. They just call it negative regulatory molecules. Nice. Well, that's good. So they can block transcription by binding to the DNA. And that's what they say about stopping it. So I'm not going to put anything in there that is extra as far as that goes, because I'm not going to add vocab to it. Let's see. So you guys, cell differentiation results from the expression of which genes are expressed in your tissue. Induction of transcription factors during development results in gene expression. We got all that. Cool. Okay, so they basically say for this is positive, so this is starting it. And then for stopping it, they just say negative regulation will sit on the DNA and not let it be done. And I mean, you can write that down, or you can just think, I mean, that would be pretty obvious. If you're blocking the DNA, if you're blocking the gene, you're not going to produce the mRNA. Okay, cool. All right, cool. Okay, let's talk a little bit about epigenetics right here, because that is regulation of this. So I'm going to make a little, here, maybe I should make a little star here too. So little bullets, promoters, transcription factors, and epigenetics. The epigenetics so DNA, I'm gonna draw a little picture for you here. So this is going to be DNA, and it's going to be wrapped around histones, because I want you to know what histones are. And we did this before. Done all this before. Okay, so in these little things here are proteins called histones. So these are histones because proteins are everywhere and do everything. And this little strand here is DNA. Okay, so your little DNA strand, which is right there in the middle being transcribed, is rolled up on proteins called histones. And it looks like a little string on a bowl, like you're just rolling it around. The histones are round, but actually they're in subunits. So you've got multiple units of histones. So they look like a little, 8-piece ball, like 8 little marbles that are in a little circle. And those histones have properties. They have methyl groups, and they have different chemicals on them that allow them to interact with other chemicals and each other and the DNA. And so all of this kind of helps your interaction. So did I spell it right? Yeah, it looks weird. So basically there's 2 things we can do. We can methylate the DNA. So methylation of DNA is one. And this is going to turn off. I'm going to put off right here. That's going to turn off transcription, and we turn it on with histone acetylation. And I'll kind of say a little bit about that, but not a lot. Okay, so methylation of DNA means we're adding a methyl group to DNA. When we add a methyl group, when the cell sends in methyl groups and they attach to the DNA, it rolls the DNA together. They actually are, DNA would be attracted to like other parts of DNA, so it pulls it shut. And if you can imagine, your little RNA polymerase cannot get in to the DNA to like express it. Okay, so now it's off, right? It's turned off, okay? Now histone acetylation does the opposite. You put acetyl groups, if you put acetyl groups on your histones, then they actually repel, the histones repel each other and roll apart. Okay, so they're like, I don't like that. And when they roll apart, it allows the DNA to be stretched open in between them. And now there's space, excuse me, for the RNA polymerase to come in. Transcription factors, promoter region. A promoter region becomes available, transcription factors sit on it, RNA polymerase is able to do its job. Histone acetylation opens it, DNA methylation closes it. I remember this because of the super fabulous, amazing example of our methylated X chromosome. Okay, so where we have 2 X chromosomes, females do. I mean, some males do too because they're like XXY. But anyway, when you have two X chromosomes, one of them will be completely methylated, all the way down to nothing, like a little squishy nothingness, okay? And it'll stay that way your whole entire life because the way the X chromosome is, you don't want too much of X chromosome produced. Evolutionarily speaking, this is getting really deep all of a sudden. Evolutionarily speaking, males only have one, and so one X chromosome produces massive amounts of protein, more than a typical DNA would, a typical chromosome would. X does like overdrive protein production. So you don't want two X's going, or else you end up with, like if you have a trisomy, like trisomy 21, it's overproduction of protein, and it makes some weird things happen. Like that's what would happen if both of our X's produced all of their proteins all the time. We would just have some strange things happening. And so one X is just methylated, completely down to nothing. And then of course there's the fun thing that you talk about that it's not the same X in every cell in your body. And so you have some X's that are producing one thing and some that are producing another, and you kind of have some different features because of that. Kind of fun, yeah.
Okay, so there was a question on the mock where it was like someone had two X, like 3 chromosomes.
Yeah, 21.
Yeah.
Just Down syndrome.
So what was like
Too much protein production. So your overproduction of proteins causes issues. Like we do not want to overproduce. That's why we have regulation. Otherwise, we just produce it all. But overproduction of proteins causes problems. Yeah.
You use the term pulls apart for histone acetylation. What exactly is it pulling?
So in acetyl group, I don't know what you know about acetyl groups. But anyway, in acetyl groups, the charge is such that whenever we can acetylate our histones, they actually will roll apart. And the histones are wrapped around DNA, like think about a string on a ball. And so if I have 2 tennis balls and I wrap string around them and set them together, if I make it where I put like magnets on either side that repel each other, it'll roll and you'll see the string open.
Like
so
amino acids.
Yeah. So, well, there's it's just like putting a magnet that's got the repelling side on the protein. So it's not technically an amino acid. It's more adding an acetyl group. Okay. So it's like, and you can add it. The cell can add it, and the cell can remove it. And so it can be temporary. So it's like open, closed. So it's a great regulation thing. And methylation is the same thing. Methyl groups can be added, methyl groups can be removed. But your X that's methylated is methylated all the time, which is why we can have a trisomy on our 23rd chromosome, and it's not really that big of a deal. Like Turner syndrome, you might see a little bit of oddities, or Klinefelters. You might see a little bit of oddities in the person, but really not. In fact, Turner syndrome only has one X. There's not even another 20. You only have, instead of 46, you only have 45 chromosomes because one X is enough. And then if you have Klinefelters, you have XXY, and those are phenotypically male, but they have just a few little differences in them because one of the X's is completely methylated. And so it's like, doesn't really matter. So it's kind of interesting. So that's how I remember which one is off, because the methylation of the X is something that's really cool to me. Like to think about, that one chromosome is completely methylated the whole entire time. And so I'm like, this is off. A whole chromosome is completely off all the time. So pretty cool. And that's another thing, Anya, if you want to look at how X methylation starts when you're a teeny little ball of cells. That's pretty cool too. Pretty cool stuff, but not in our standards. So we'll move on. Methylation of the X is in our standards as far as that goes, just as being as epigenetic change that stops the transcription of the X. Okay, let's see if I hit all of the transcription stuff before we move on. Let's just make sure I don't have to go back and add anything. Oh, see? I almost forgot. Good thing I looked. Operons. What the muffin top am I supposed to put operons? So let's see. Prokaryotes. Do you guys have space under enzymes?
Yeah.
Okay, maybe I'll just do a little prokaryotic thing under here. So I'll just put mine up here though, so that you can see it. But because I'm going to want this for the rest of the regulation of transcription translation overall. But let's do a little special notice to prokaryotes. So let's do some differences in prokaryotes, and I'll just add to this as we hit differences along the way. Okay. And so prokaryotes. So here's your bacteria and stuff, and so you can tuck it wherever you can fit it. But basically what we have here, so let's talk about transcription. So obviously transcription is there's no nucleus, so that should be obvious to you. And so transcription occurs just in the cell, in the nucleoid region, middle of the cell. Okay. For this regulation, we don't see this, not in prokaryotes. Okay. We don't see epigenetics because they don't have histones. So that's kind of funny. So they don't have their DNA is not wrapped in histones. And so they don't have any of the histone or methylation or any of that kind of stuff. They do have promoters, but not the same kind. So what we see here then for our regulation is operons. And operons are just a series of genes. So let me draw one here. See if I can remember everything. Geez. This is a lot to keep in my brain at one time. It's kind of strange.
I thought eukaryotes are the only ones that have like the operons exons.
Operon is not an exon. Operon is like promoter region. It turns on all the genes. Regulation. We're going to get to exon and introns in a second. Make sure you don't get those ending ons confused. Okay, operons are just a series of genes. So usually it's multiple genes. If I can spell, multiple genes in a series is kind of what they are. That says in a series. That's hard to read. I apologize. So basically, you'll have we typically box in regions. Now, no, there's not boxes. I'm just saying this area of the DNA right here. So this is your operon. Is just like regions of DNA is what it is. Okay, and so it's like a little area of DNA. Now, remember, this is not a linear chromosome, right? This is one circular big chromosome, and we're just in one little part of it. I mean, if you wanted to draw out the whole thing. But just keep in mind, this is prokaryotes, so circular DNA, right? And so basically you have your place up here where you're producing your repressor. So this is your repressor gene, or the regulatory sequence, if you want to call it that. Okay, so there's your regulatory spot right there. It's going to be way upstream, okay? And then you're going to have your promoter region, because they have promoters too. And then you're going to have your operon. This is where your repressor will attach if you have one. And then you'll have your structural genes. And let's just draw a couple. I'm going to draw 3. This is gene X, Y, and Z. I don't know why we always use the end of the alphabet for these. They always do, though. Kind of funny. Okay, so X, Y, and Z are the actual target genes. They're like, hey, those are your genes. This is what you want. So those are your genes that you're targeting. You want mRNA produced from those. Notice there's 3 in a row. This is why it's an operon. An operon typically has multiple genes in a row. And they're going to all make mRNA where the proteins kind of work together. And so it's like a one-stop shop. Like you can turn on the gene sequences and you get everything you need to do the work, whatever the work is. Okay, and so you've got your repressor being produced here, and then your promoter. This would be just like on a eukaryote promoter. This is like the spot where you would have your polymerase attaching, and then your operon is where, if you have a repressor produced from your regulatory sequence, I would lock on right there. Oh no, now the polymerase can't get by. It's stuck, and so now it won't be turned on. Or maybe the promoter comes off. The repressor comes off, and now it's like, yay. We can make, and that's it. Like operons are actually pretty easy. Once you figure out what the heck is going on with them, it's like it's either stopped because you've produced a repressor and it's stopping it, or there's no repressor and it's going. Now, the fun thing is figuring out if they're inducible or repressible. Okay, and really that's not that hard either. Okay, so they're either inducible or repressible. If they're inducible, then they're off all the time. Off typically. Because now you can induce them. If they're repressible, they're on typically. So things you need some things the little prokaryote needs all the time, like amino acid production to make proteins, are going to be repressible, because they should be making those amino acids all the time, because it needs proteins all the time. But if all of a sudden there's a buildup of those proteins, it's going to turn off. And it'll be like, whoa, too much. Don't make any more. You're wasting energy. And we don't have room for any more anyway. And so then it will be repressed. But but usually it's on. On the other side, inducible, this is your lac operon, or your lactose one. Inducible is like, oh, we typically leave that one off because we don't need it. But, oh, dang, here's some lactose, and we're fresh out of glucose. We better turn it on. Okay, so that's those. Cool. Questions? It's really not too bad. Okay, so then moving straight along, Natalie mentioned the next step, which is mRNA processing. This only occurs in eukaryotes. So we have our, so introns are cut out. Introns are out, and exons are in. So this is the expressed part, exons. It's expressed, so we call it exons. And then introns are just little things in the middle that need to be chopped. Okay, so they are cut out. So this is mRNA processing. Make sure mRNA is shorter.
What is this translation?
This is moving in between. This is still in the nucleus. We are still in the nucleus. So this is not, we're not quite done making our mRNA until we get it processed. Now it's done. Oh, wait, no, it's not. We have to cap it. 5' cap poly A tail. 5' cap and poly A tail. Poly A tail's on the 3' end, and it's got just a bunch of A's. And we're not exactly sure why they do that. We think maybe to preserve the mRNA as it moves through the cell. We got to get translation done in 6 minutes. Let's go. Okay. So once it's processed, now we're going to move to translation. I hope you have some space right underneath to do quick translation talk. I'm going to erase a little bit because I'm telling you this is full of crap. What should I erase? I'm going to erase this. If you don't have any room, flip your paper and put translation on the bottom. I mean on the back. Okay, so we've got our translation. Now we're moving into translation. This will go a little faster because there's not as much regulation of it. So first of all, we are trying to translate. So now this is going from mRNA to protein. mRNA is going to be translated to protein. You'll use your codon chart to do that. There will be a codon. I can almost guarantee you there'll be a codon chart on your test somewhere. It'll either be asking you about mutations. Oh my gosh, the DNA's changed. What do you get now? You know, something like that. Okay. This is going to the ribosome, right? So we have our ribosome. So what do you need to know about that? rRNA equals ribosome. Okay. Of course, there are some proteins involved in that, the way it's balled together. But basically, if you have a mutation in your rRNA, you have a mutation in your ribosome. Because rRNA is produced in a lot of the same way that mRNA is, right? It's from a DNA code, and it's produced from that. So if there's a mutation in the DNA that affects your rRNA, then you are having a mutated ribosome. Now, your proteins probably won't be formed exactly properly for at least those types of ribosomes, okay? So what could keep translation from happening? Okay, well, let's go over here. And let's say, I call them siRNA. But you can also call them micro RNAs. You can call them small RNAs. Basically, AKA, whatever. They're not exactly the same, but we use them as if they are the same. Like they have different roles or how they work, but we are just talking about them in general as an umbrella term. If you see small RNA or microRNA or siRNA, si just means small interfering. And that's why I like it, because it literally tells me what's happening here. This will stop translation, okay? This will lock onto the mRNA and stop translation. So we've got siRNA, we've got rRNA, we've got mRNA, and the last one we need is tRNA. So here's our ribosome. Here should be, okay? And it's ribosomes are 2 subunits. Here's our mRNA. The last one that we need is the tRNA, which transfers amino acids in. So this brings amino acids in. I'm going to abbreviate it AA for amino acid. And I don't know how you want to draw it. They usually draw them something like this. And then they'll have, I have to think, AUG. Okay. So they'll have a anti-codon. That's a word you have to know. Okay, that'll match up with a codon. So this one's matching up with AUG, which is your start codon. And then you'll just go one codon at a time. until you produce your protein.
What does that say? Brings what in?
Amino acid, AA. I just call it AA because it's easier than writing amino acid every time. Okay, anybody need me to read any of that parts to them that they can't really see it? It's kind of a mess because I'm hurrying. And then finally, I think the only thing left, we didn't do biotech. I'll hit that at some point. I'll hit that when we do the math. The only thing I think that we need to make sure that you know is that the one thing that I've seen them catch students on more than once is that methionine, the start codon. So this is UAC. It would code for AUG. It is a start codon, right? Methionine is the start. You know that? Sometimes they will have a string of DNA and you're not supposed to start coding until you hit the start codon. It'll even say, begin with the start codon. And so you have to look for it, right? AUG, methionine. Okay, methionine brings an amino acid in. The stop codon does not. And that's something that I've seen them catch kids on a lot. And basically they'll say, how many nucleotide, bases or whatever did it take to build this protein? And then they'll have like the code, like AUG, blah, 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 blah. Students will forget that the stop codon does not code for codon. It doesn't code for an amino acid. There's no amino acid for stop, okay? It's a release factor. It's just a protein that comes in and goes chop, okay? And so there is one for your start, but not for your stop, okay? So just kind of keep that in mind as far as, I don't know if they'll have you do it. They've had to do it in free response questions. They've had to do it in multiple choice questions. I've seen it multiple times where they want to know how many nucleotide bases did it take to make this protein, okay?
How many would it be?
Well, it would just depend. It would be one for every 3 letters. AUG is 1 amino acid, blah, blah, blah. Next 3 is 1. And then when you get to the stop, you don't count it. So that's the thing that they trick. That's the place that they get you. And actually, they awarded 1 point if you left it off, but they awarded 2 if you knew that the stop codon didn't. So it was like a bonus point almost. Okay, I'm sure I forgot some stuff because that's a lot. I'll pick it up where we can in the